This is why it's good to have a friend who's an IT expert. Oh, you named it. Nice.
Thank you.
Oh, you're welcome.
Yeah.
And so then how do I send?
Copy the link to them. This link will work. It's fine.
Awesome.
It's just taking them to YouTube.
Can you send me a link? I can double check with mine.
Oh, wait. I got it.
This is so exciting. And super cute. Do you want like cinnamon rolls?
I'm good. I can search this on YouTube because it's not popular.
I think I got it.
I got it. I can double check on mine.
What's your phone number?
I got it. I can just search you.
Oh, really?
Ooh, shady. I like it.
Okay. My sister can see me.
Yay.
Can you hear me?
Do you want to move the microphones? They're not all pointy.
I do notice that the public internet is actually really slow. So
Okay.
I may ask Alex to log in. If we ask her if she Danny know what's going on.
Okay. Love you guys. All right. Okay. Thank you. I don't know how to pay you back except for give you a big hug after this.
Sure, do
Can I
that.
just have a big hug?
Yeah.
Wait.
That was just a mini panic attack I just had. Thank you.
You look so pretty and so professional.
Okay. Oh, yeah.
I can adjust this for you a little bit. Hey Jenna, come see it. Where you want to put this on?
Hey, you just make me look cute.
I know. Is that good? Go over there. I take the phone with you. Hey, I can help you adjust the camera angle.
I like your stickers.
Yeah.
Those are cute. Do you not want food?
You might want to be able to see the screen. Yeah. He's amazing. You should keep him forever.
You don't want to put on first row? Can do the. If you put this on the first row, you can skip the chair part and you can see the whole poster. If you want to.
I like a little bit of distance.
Okay.
Sorry.
Thank you. Don't block my camera.
I so enjoyed all that last night. I'm like,
No, you're not even doing it. Where is it?
Sit right there. If you don't sit right there, I'm
Right here.
offended. Say hi to Mom and Dad. I'm always offended.
Can you see his wallet? Yes, you do.
They're good?
I don't know.
Doing it. No, you're fine. You do your thing first, and then I'll.
No thanks.
Okay.
Yeah.
I mean, she just like texted me randomly and was like, "Oh yeah." Cool. So I'm not like finalized yet.
Hey, how's it going? Good morning.
No, I
How are you doing?
Good.
See you again.
Yeah. Good idea.
Sorry. I'm just gonna. Okay. No, you're good. You're good. No, I'm sorry. Oh God, I didn't mean to do that. That's okay. It's okay. I'm sorry. I'm the worst. It's fine. I volunteers tribute. It's okay. Oh. This is how you ask questions. You just scream into the microphone.
It's like many times from like a crowd who, like,
You can be like,
the senator from Ohio.
yeah, exactly. Senator Jacob,
Oh, cool.
representative from Dubai. You'll meet them later. Sure. These are for you and Chris. To open after your dissertation.
Okay. So we should.
It's filled with raisins. I mean, I guess you could, but don't.
Good, but
The last word. I'll know. I'll know if you open them early. Good. Okay, Alice.
Did she change her last name?
No, it's Anthony G. Johnson.
Yes, that's it.
She has two last names. Because in Ohio, you can have two last names. So doing that. And then if you move to a different state, they have to.
Honor that?
Yeah, they would. They have to honor it. Yes. If you move to another state. Oh my fucking god, I'm doing it. Okay, sorry.
What's your last name, Blondie?
My last name is Kosmission currently, but it's gonna be Kosmission Maggie. That's my goal. Yeah, that's right. It was very much like, can you just pick a name? Like, I really don't care if it's mine or yours you keep, but that's really fun. Were you gonna hyphenate it? Because that sounds fucking miserable. Yeah. How would you put that on any form? I wouldn't. It would never fit. Mother-in-law. And so that's why the two last name idea sounds great. Because then I can pick.
I saw about changing my name to Dragon Slayer.
Don't do that. First of all, it sounds just
Yeah.
Why is it racist? How dragons?
Yeah, that's my thinking. Is like,
Do you know what kind of Chinese or what kind of Asian I am?
and, you know, when like registering in schools or whatever. But then professionally, I could keep the mission and wouldn't be like rude or wrong. So,
Like I think technically racist was being because I guess which actually she didn't need to. She thought she like officially changed it, like she didn't change it with like the right people. Because it's like on a lot of like websites it was like changed, but then like I guess with like the actual like grammar school, it wasn't like all these like like stuff like.
I think it's hard.
I don't know, yeah, I feel like super hard. Like it was like, and it was like what she was like trying to get her like dissertation for format check. Like they were like, oh yes, like just put your name on your
Yeah, exactly. Especially like the moment where like you need to like pay money to like change some letters. I like this, you know. Yeah. No, I'm not thrilled about going to the social security office.
I should get on my eyes.
Like,
Like
I have eye drops. Yeah. Everyone makes it sound like it takes a second, but it takes
and it
forever.
really sucks because they don't want you to do stuff with your family. Because I mean, like,
Yeah.
you're like, so they see this, I'm not. Yeah, you can bring.
I'm so bored.
Can't open
It's so bad. It's like they're trying to kill you with boredom. It's like a test. Do you really want to change your name? Well, I just give you my job.
No, I like this better.
I have a napkin if you want it. Ooh, cute.
Let's get started. I'm very excited. I'm Jibu, the mentor of Jenna. Very happy to introduce Jenna today. And first of all, thanks everybody for coming, particularly committee member and Erna and Jessica. Also, we have graduate school representative, Dr. Federica, is coming for support and host the exam later. As just the format, we will have a general seminar about 45 minutes around, and then we open to questions from general audience and public. I think it would be better you save your question after Jenna's talk, and then committee members and Federica, we're going to get together close door meeting after your seminar. I think everybody knows Jenna, and I don't think I have to introduce her because I think Jenna knows more people than I do. Even I mean, was longer than Jenna. And she has been in the lab for about 4 years, joined OSU 5 years ago, and graduated from Stanford University in Boston in 2013. And Jenna did a really good job in the lab working on this very interesting host enzyme called the sandwich one. I leave all the topic and the science part for Jenna to introduce, and also today we are very glad to see Jenna's family also coming. But I do want to share a very personal story. I remember about 4 years ago when Jenna finished her rotation. I think we were very excited talking about her potential fitness project and join the lab. I think really after 4 years work, Jenna solved a lot of key questions. I mean, real key questions like this. Because 4 years after her rotation, we were really excited about the ongoing project. We were talking, and then she left. After my work, I couldn't find my key. The whole set of the keys somehow missing. And I called my wife. I couldn't drive, and she picked me up. And the next day, I was just thinking almost the whole evening, where's my key? And then I realized that we were talking, and Jenna had a big backpack, and she left very excited. After talking the project, I said, maybe there's a chance that Jenna grabbed my key. So the first thing, that next day morning, I rushed to the lab and her office trying to find Jenna. Of course, Jenna was not there. She was on the class at some course there. And I left a message and emailed her. I said, Jenna, could you please check if by any chance you had my key in your backpack? Indeed. She solved my key question, and this is topic today because one of the most challenging questions in the SAMH1 research field is what's the fundamental mechanism. I think we're going to hear this story. And the last thing is, Jenna will be graduating very soon. We will all miss her, particularly myself, because in the last 2 years, Jenna has been my teaching assistant for a very extensive biology course. That in 2 years, and very much appreciated Jenna's help. Actually, that made the whole course much easier, and many faculty members participated part of the course. Really grateful Jenna's lot of help. So I think I will stop here and leave science part for Jenna. Thank you.
Firstly, thank you all for being here. I'm really excited to share with you the bulk of my dissertation work. A lot has gone into this project because I honestly really loved this project and the opportunity to be a member of the lab and to really be given the opportunity to explore these really big questions. Today I'm going to talk about the mechanisms of HIV-1 restriction by SAMH1. By the end of this, you guys will almost be an expert in SAMH1. Firstly, just an overview of my talk so you know what I'm getting close to the end. First, we're going to go through an introduction to HIV, AIDS, and SAMH1. And then the first part of the talk is going to be about trying to discern which enzyme activity of SAMH1 is essential for HIV-1 restriction. And this part of the project was born out of a little bit of controversy and really was a joy to work on. And then secondly, we're going to talk about a role for SAMH1 in modulating HIV-1 gene expression, which is a novel role for SAMH1. And then we'll summarize our findings and talk a little bit about future directions. So firstly, AIDS, HIV-1, and SAMH1. So HIV-1 is a causative agent of AIDS, and AIDS is a disease that affects millions of people worldwide. About 37 million people have died of AIDS thus far since its discovery in the 1980s. Now, although things are getting better for patients infected with HIV, for example, AIDS-related deaths have decreased by 400,000 in the last 15 years, there's still a lot more work to be done. And the reason why this number has decreased is due to a lot of factors. Firstly, it's due to the use of antiretroviral therapy. It used to be that you had to take a cocktail of drugs, sometimes 10 pills a day, to keep your infection under control. And now it's down to 1 pill. And as you can see, because of this, because of the increase in treatment, we've gone from less than 1 million patients on antiretroviral therapy to 17 million. The goal is to have every person who's HIV positive in antiretroviral therapy within the next, by 2020, that's the target. And another thing that has decreased the infection rate is education. So understanding how people get infected with HIV and also how HIV progresses to AIDS. But today we're not going to talk about AIDS. We're going to talk about HIV. We're going to go into a little bit detail into the life cycle of HIV. And by understanding the life cycle of HIV, we're able to not only better understand the virus itself, but actually better understand our own immune system. So HIV is a retrovirus. This is a, in my opinion, very special type of virus that once it binds to a primary receptor and co-receptor, it fuses with the cellular membrane. It has to reverse transcribe its single-stranded genomic RNA into double-stranded cDNA. And it does this through the incorporation of host deoxynucleotide triphosphates or DNTPs. I like to explain this to people as you need the Legos to build a Lego tower, and the DNTPs are the Legos that build this Lego tower. And once you have this double-stranded cDNA, it can be integrated into the host genome, into the host chromosome. And this integration is one of the causes of long-term and persistent infection, because at the moment, it's impossible for our bodies to excise this proviral genome effectively to clear the virus. But after integration, you can have transcription of viral RNA, either full-length genomic RNA that will then be packaged into newly synthesized virions, or you could have production of mRNA and viral protein. Now, understanding, as I said, this viral life cycle helps us understand the virus. And the most important reason for understanding the virus is to target it. And the way we target it is through therapies. And we have antiretroviral therapies that target binding, fusion. We target reverse transcription in two ways, either with a nucleoside analog RT inhibitor or a non-nucleoside analog RT inhibitor. We have integrase inhibitors, and we have maturation inhibitors, protease inhibitors. So by targeting the virus at these various points, we can stop it in its track. But you can kind of see that there's a little bit of an empty space here, where we don't have current antiretroviral therapies to prevent translation of protein and transcription. But something amazing happens in our bodies when we're infected with viruses over millennia. And that's the development of restriction factors. And restriction factors are interferon-stimulated genes in our body that respond to infection and actually block infection at various points in the life cycle. For example, apocrine proteins are cytidine deaminases that cause terminal mutations in the genome during reverse transcription and block infection at that point. We also have tetherin, which actually, it's a great name. It tethers the virion to the surface of the cell membrane and prevents release. But today we're going to talk about SAMH2, this amazing protein that blocks HIV infection at the step of reverse transcription. And let's go into a little bit more detail. So I talked a little bit about those Lego blocks, the DNTPs that are required to reverse transcribe HIV1 genomic RNA into cDNA. Now, in certain cell types, if the DNTP levels are too low, you see a block in infection. And for a long time, scientists were noticing that certain myeloid lineage cells and quiescent CD4 T cells were reticent to infection. They couldn't be infected. They also noted that these cells had really low DNTP levels. And if you added exogenous DNTPs, so if you added DNTPs from a different source, that you can rescue infection. But in 2011, an amazing discovery was found that SAMH2 was the protein responsible for reducing DNTP levels in the cell, thereby blocking infection at the step of reverse transcription. Now, a little more detail about SAMH2. SAMH2 exists as a monomer, dimer, tetramer, and it tetramerizes through allosteric binding of DNTPs. So first, DGTP or GTP will bind to the first allosteric site, and this will cause dimerization. And then any DNTP will bind to the second allosteric site and induce tetramerization. And only the catalytically active tetramer is able to hydrolyze these DNTPs. So as you can see, there's a lot of structure-based control of this protein, and I'll go into a little bit more detail about that later. But SAMH2 is a 626 amino acid long protein that has, you guessed it, a SAM and an HD domain. The sterile alpha motif is known traditionally to be a nucleic acid binding domain or a protein-protein interacting domain. A little upstream of that on the N-terminus is a nuclear localization signal. SAMH2 is primarily nuclear localized, but it is a shuttling protein. So we do see it in the cytoplasm. Then we have the HD domain. And this HD domain is essential for HIV restriction because of the HD residues, 206 and 207. These residues are in the catalytic core, and they are responsible for the hydrolysis of the DNTPs. Then we have this phosphorylation site at threonine 592, and this is incredibly important because our group and others have shown that phosphorylation at this site causes SAMH2 to dissociate from its tetrameric form. And when SAMH2 is a monomer or a dimer, it can't hydrolyze DNTPs. So it appears that phosphorylation at threonine 592 is a way to negatively regulate SAMH1's activity. Then lastly, we have this VPX interacting domain. I'll talk a little bit about VPX in a minute, but VPX is the protein encoded by HIV2, not HIV1. But VPX will interact with this VPX interacting domain and stimulate SAMH1's degradation by the ubiquitin ligase complex. So it's a tool that we can use in the lab, although you won't see it in any of the data I show you today, to knock down SAMH1 expression in cells. So as I said, SAMH1 is a DNTPase. It's actually the only identified mammalian DNTPase, and that way it works opposite of ribonuclease reductase that produces DNTPs. And when DNTP levels are high, we see high infection because HIV has access to all these DNTPs to make its cDNA. However, when SAMH1 works and it hydrolyzes these DNTPs, DNTP levels are low, we have low infection or blocking infection. And today, in the first part of my talk, I'm going to mention two important mutants. H206R, D207N, I'm just going to call it HDRN, and threonine 592A. So HDRN, we took these HD residues and mutated them, and this enzyme is enzymatically inactive. It still forms stable tetramers, but it cannot hydrolyze DNTPs. So we use this as a negative control. The threonine 592A is a phosphoablative mutant, so it can't be phosphorylated. So in certain cell types, since it can't be phosphorylated, we hypothesized that it would be active, constitutively active in comparison. And that's summarized here. No restriction with HDRN, but we see restriction with threonine 592A. And like I said, SAMH1's activity is controlled by structure. Not only can SAMH1 be phosphorylated and prevent this tetramerization, there can also be oxidation and production of a disulfide bridge that prevents tetramerization. And then lastly, although it's not a post-translational modification, it's been shown that if SAMH1 binds to single-stranded nucleic acids, this binding occurs at the dimer-dimer interface, and it also impairs tetramerization. So these are just an example of 3 ways that SAMH1 can be negatively regulated. Its potent DNTPase activity can be negatively regulated. Additionally, SAMH1 is involved in other cellular functions. It's phosphorylated at that threonine 592A by cell cycle-dependent cyclins. And this makes sense if you think about the cell cycle. When do you want high DNTP levels? When you're replicating your DNA. When do you want low DNTP levels? When you don't want your cells to be proliferating. So SAMH1's activity is regulated throughout the cell cycle to modulate DNTP levels. Again, SAMH1 restricts HIV infection and also has a role in the innate immune response, although I won't be going into detail about that today. So to get into the first story, I told you about this amazing model where SAMH1 reduces DNTP pools, and that leads to a restriction in HIV infection. However, in 2013, a paper came out that showed that actually SAMH1 is also a nucleic acid binding protein. Not only can it bind to single-stranded RNAs and DNAs, but it can bind to in vitro transcribed fragments of HIV1 GAG RNA. So this paper hypothesized that maybe SAMH1 has a nuclease or ribonuclease activity that may be contributing to HIV restriction. So for example, if SAMH1 was binding and degrading this genomic RNA, we wouldn't see any downstream infection. At the same time, SAMH1 could have been degrading newly transcribed genomic RNA or mRNA, thereby also blocking infection and production of newly synthesized virions. So the hypothesis was that SAMH1 exerts a ribonuclease activity against HIV RNA, thereby blocking infection. Then, actually, I was really excited about exploring this, and I went on my honeymoon. And while I was on my honeymoon, this paper came out and just totally ruined my life. Ended up being great, though. Spoiler alert. So, a paper came out in Nature Medicine that really tried to prove that SAMH1 is a ribonuclease, and that's how it restricts infection. So, to walk you through this data, we have DCTP levels here, and these are cells that are either that are expressing wild-type SAMH1. We're only going to talk about wild-type first. And so you can see SAMH1 reduces DCTP levels. We know that. SAMH1 is a DNTPase. SAMH1 blocks infection. We already knew that. But interestingly, when they measured HIV1 RNA levels, there was also a dip. And so this group thought, okay, so which enzyme activity is responsible for this block in infection? Is it because it's reducing DNTPs or it's reducing HIV RNA levels? So let's just ignore some of these mutants. So they developed 2 point mutations that they said delineated the RNase and DNTPase activity. So firstly, this D137N could not reduce DNTP levels, but could restrict infection. So that's interesting. You're telling me that this enzyme does not reduce DNTP levels, but it's still restricting infection. When they measured HIV RNA levels, they saw that it reduced RNA levels. So this is when they made the preliminary conclusion. And then they developed another mutation, the Q548N, that reduced DCTP levels, so it had a DNTPase activity, but did not restrict infection. Additionally, it did not reduce RNA levels. So, by using these mutants, wild type, they say has an RNase, DNTPase, and restricts, but by using these 2 mutants to tease apart these activities, they came to the conclusion that since the D137N could not reduce DNTPs, but could reduce genomic RNA, that it was the enzyme activity responsible. And we thought, okay, this is really interesting. Let's confirm it and do additional assays to really characterize this activity. So what we did first was we stably expressed these mutants and wild type in U937 cells. We did this by lentiviral transduction. Now, the first step we had to do was to make sure that these cells would differentiate into macrophage-like cells. We wanted something that was physiologically relevant. So first we stayed with CD11B. This is an MDM control, so you can see the SAMH1 levels in MDMs. And we were able to achieve about 90% activation upon PMA treatment, which is to be expected. And then all 4 of our cell lines were able to differentiate into macrophage-like cells. So that was step 1. Step 2 was to measure whether these mutants could restrict HIV infection to the level of wild type. So we infected these U937 cells with an HIV reporter virus, and we were able to see that all 3 SAMH1s were able to block infection. Now, we used nevirapine, which is a reverse transcription inhibitor, as a control to make sure that this restriction was happening at the step of, reverse transcription. So this is one of our controls. Additionally, we went ahead and measured incoming genomic RNA levels by qPCR. So if we look at our handy graph, we're measuring the levels of this genomic RNA. So you would expect if SAMH1 was degrading genomic RNA, we would see an effect. All 4 of our cell lines had similar levels of genomic RNA, indicating that SAMH1 is not degrading this. And you'll see this characteristic decrease in genomic RNA as time progresses, as that genomic RNA is used as a substrate to make cDNA. So our conclusion was SAMH1 expression does not affect HBV1 genomic RNA levels in these PMA-treated U937 cells. Next, we wanted to measure late reverse transcription products. Now, we would hypothesize that if SAMH1 is blocking infection at the step of reverse transcription, we would see an effect in cDNA levels, and we do. At 12 and 24 hours, all 3 SAMH1 expressing cell lines are able to have reduced late RT products upon PMA treatment. And obviously the vector cells with no SAMH1 expression, we use as a control, as our vector control. So as I said, SAMH1 does not affect, SAMH1 expression does decrease cDNA synthesis, indicating to us that the restriction is happening upstream of that cDNA synthesis. So we directly measured DNTPs. We took these cell lines, PMA treated them, so they expressed SAMH1 to high levels, as I showed you in the first figure, and then we extracted the DNTPs for analysis. And all 3 cell lines had reduced DNTP levels. So then we want to try a different assay. We were curious if SAMH1 might be having an effect on newly transcribed mRNA, protein, and the newly synthesized virions. So we used a co-transfection method. By transfecting in a proviral genome, we totally eliminate the need for DNTPs because you're not producing any cDNA. You see transcription off of plasmid. And then we also co-express SAMH1. And this is in 293 T cells. We use this cell line as a virus-producing cell. So we co-transfected. We actually collected the virions, and we did 2 things. Firstly, we lysed the protein in the cell. This is what the Western blots will look like. We wanted to look at GAP protein synthesis in the cell. We also lysed the virions. We wanted to see what the P24 levels were with SAMH1 expression. That's a readout of whether or not enough viral protein is being made. We also took these cells and infected a ghost cell. A ghost cell is a human osteosarcoma-derived GFP reporter cell line that expresses GFP upon infection. So we're measuring three things, viral protein production in the cell, in the virion, and then also if infectivity is bothered. Now, we would expect infectivity to be reduced if SAMH1 is degrading genomic RNA, because no further infection can happen. So as I said, this assay, this is a Western blot, is used to measure this protein production. So if SAMH1 was degrading mRNA, we would see an affected protein. And we use the threonine 592A, that constitutively active protein, because in 293 T cells, SAMH1 is phosphorylated. I just want to note, though, that the DNTP levels in 293 T cells are really high. And as you can see, as we increase expression of SAMH1, there was no effect on GAG, which is a major protein produced by HIV. And there's also no effect on P24 in the virions. So the virions appear to be healthy. Well, like I said, you never know until you do an infectivity assay, which we did. And viruses produced in the presence of SAMH1, either wild type or phosphoablative, were completely healthy compared to virions that were produced in cells that didn't express SAMH1. And that indicated to us that SAMH1 expression does not affect HIV-1 protein production or infectivity in 293 T cells. Then we really were interested, okay, so it's not affecting mRNA, doesn't seem to affect genomic RNA, but would it affect an RNA virus? So this is one of my favorite David Baltimore classifications of all the virus, but don't worry, I'm not going to give you a lecture. HIV is a retrovirus that, as I told you, has to reverse transcribe its genomic RNA into cDNA, and it needs DNTPs to do this. But there are RNA viruses, like influenza A and Sendai, that don't need DNTPs. So we hypothesized that if SAMH1 was degrading the genomic RNA of these RNA viruses, we would see an effect in infectivity. And also, just as an additional fun control, influenza A replicates in the nucleus and Sendai in the cytoplasm. So we had all of our bases covered. And what we're able to see is that SAMH1 expression had no effect on Sendai or influenza A virus at 2 MOIs, but these proteins potently suppressed HIV infection. So SAMH1 cannot restrict minor-stranded, single-stranded RNA viruses, indicating to us that SAMH1 does not act as a ribonuclease. So you may be asking, well, what about that other paper? They showed that it has ribonuclease activity. What's really going on? And so in a collaboration with the Zhang Lab at Yale, we were able to do an experiment where we purified 2 batches of recombinant SAMH1 protein. Done side by side, very stringent. Here we have our elutions. And then using a nuclease activity, they were able to see that in only one sample, sample 1, does SAMH1 have that very characteristic cleavage pattern of this FAM-labeled nucleotide. Indicating to us, since we only see the nuclease activity in one batch and not the other, that the nuclease activity is likely due to bacterial contamination during the purification process. Now, since we were able to produce this data, many papers have confirmed that SAMH1 maintains no active site nuclease activity, and that and a few groups have confirmed as well that it's likely due to bacterial contamination. So make clean preps is the lesson story. So moving on. Although we were able to prove that SAMH1 isn't a nuclease, we couldn't disprove that it binds nucleic acids. And the function of this binding was still really unknown. So I'm going to tell you a little bit about the project. I know this is a little bit of a spoiler alert about how we went on to explore what this binding effect might, the purpose of this binding effect might be. So firstly, a little crash course in latency. When HIV, when you have a naive CD4 T cell that is activated by a dendritic cell or an antigen, what happens is these cells become activated and they proliferate. And this is a part of our immune response. Now, these cells, eventually, some of them will die, but some will also become memory precursor cells. And eventually, some will become resting memory CD4 T cells. Now, when these activated CD4 T cells, which, in case I wasn't clear, these cells do have SAMH1 expression, but when it's activated, SAMH1 is phosphorylated. When we have activated CD4 T cells that are able to be infected by HIV, you have that whole process of the life cycle, reverse transcription, integration, gene expression, and one of two things will happen. Either the cell will continue to produce virus, but eventually it'll die from cytopathic effects or by immune clearance. Now, there is a chance that this virus can infect memory precursor cell. So the cell is just active enough to become infected, but then it transitions into a resting memory CD4 T cell. And when this happens, again, one of two things can happen. Either if it remains active enough, you could have production of virion, or if it becomes completely transcriptionally silent, and now SAMH1 is highly expressed in these cells, I'll show you evidence in a second, you can have a latently infected cells. And these cells are very long living and can be activated at a later time. So you can take this latently infected cell, treat it with a latency reversing agent, and it will produce virus. So as I said, SAMH1 is expressed, so these two chemicals right here, CD3, CD28, PHA, activate resting CD4 T cells. And you see SAMH1 is expressed in these cells, highly expressed in resting CD4 T cells. Also here we have the relative mRNA levels, and you can see that SAMH1 mRNA is also detectable in these cell types. So SAMH1 is highly expressed in these cells that contribute to latency, or a huge component of the latent reservoir, but no one has ever really tried to figure out if SAMH1 may have a role in latency. So just a little bit more information about the latent provirus. Here we have the entire HIV genome. And what's most important for this project, for my project, is this 5' LTR. The LTR is the promoter. It is responsible for enhancing or suppressing LTR-driven gene expression. And I've sort of blown it up here to show you just a handful of transcription factors that bind to the LTR region that help regulate its activity. So this is a very complicated control system, and it makes sense. The virionic times, it's a balancing act, because if you're producing a lot of virus, you're also producing a lot of material that can be sensed by the immune system. But also you don't want to completely suppress transcription because you won't make any new virions. And so just another piece of data showing that SAMH1 is able to bind to DNA, single-stranded DNA, in low levels of double-stranded DNA. So we have these two observations that SAMH1 is a nucleic acid binding protein, and also that it's expressed in cells that contribute to the latent reservoir. So we were curious whether SAMH1 might be affecting the LTR. Now the first thing we did was use an LTR-driven luciferase construct. So we took that promoter upstream of a luciferase gene, and any activation of that promoter would lead to luciferase activity that we can use as a readout. And as we expressed SAMH1, we saw suppression in this LTR-driven luciferase activity. As a transcription control, we also used HSV TK-driven ranilla luciferase construct, and SAMH1 had no effect. We also measured the mRNA of luciferase, and as we can see that the decrease in mRNA levels correlates to the decrease in activity, we made the conclusion that this suppression is at the level of transcription. But we were also curious, okay, that's when we overexpress SAMH1 in 293 T cells. What about endogenous SAMH1? Can endogenous SAMH1 suppress LTR-driven gene expression? So we used a well-characterized THP1 cell line that CRISPR-Cas9 was used to produce a knockout of SAMH1. And we nucleoaffected that LTR-driven luciferase construct and the TK-driven ranilla luciferase construct into these THP1 cells. And when we removed SAMH1, there was an awesome enhancement of LTR-driven gene expression. No effect on the TK-driven ranilla luciferase construct, suggesting to us that this suppression, or in this case, enhancement, is specific to the LTR. So just to kind of summarize so far, when SAMH1 is expressed, we see a suppression in LTR-driven gene expression, and when we remove SAMH1, we see an increase. But we wanted to look at a few mutants. We're going to use the HTRN and the threonine 592A, which I know you're all experts in. So we have the constitutively active and also the enzymatically dead. We were interested, is phosphorylation involved? And also, is the DHPase activity of SAMH1 essential? And we expressed these proteins in 293 T cells. And while the mutant was able to suppress LTR-driven gene expression, wild type was able to suppress LTR-driven gene expression, the mutants had an impaired ability to do so. And we were interested why this could be. So we used a JLAT latency model. So up until this point, we've been using 293 T cells, THP1 cells, but we really wanted to look at the integrated proviral genome. And JLAT cells are derived from Jurkat cells, and they've been infected with this HIV genome. There's no envelope, so you're not producing any new virions, and there's a GFP in the NEF region. So any activation or transcription off this proviral DNA will lead to expression of GFP. So what we do is we transduce the JLAT cells with P-lenti-SAMH21, so we make a stable cell line, and then we treat the cells with LRAs, which are latency reversing agents, and then we measure GFP to see if there are any changes upon expression with SAMH21. So firstly, we expressed SAMH21 in these cells. We got similar expression, and we measured GFP upon treatment with PMA and iodomycin, which is a potent activator of the LTR in these JLAT cells. And we saw that wild-type SAMH1 was able to suppress activation of transcription. So we decided to determine if this was due to binding. So we IP'd out SAMH1, eluted all the DNA, and did a qPCR experiment. So we call this a CHIP qPCR. And we used primers for our qPCR that scan the whole genome, including GFP. It's a good control because it's a non-viral gene, but it's located in the genome. So if we saw any non-specific amplification, it would be a good measure of SAMH1's binding non-specifically. And then when we use these primers with threonine 592 and HTRN, we see that only wild-type SAMH1 is binding to the genome, and it only binds to the LTR. So our mutants are unable to suppress reactivation, and they're also unable to bind. So this was our first evidence that the suppression of LTR-driven gene expression by SAMH1 might be due to binding. So to really characterize this binding, we did fluorescence anisotropy binding assays. And these assays are based on the observation that an oligonucleotide tumbles through space differently when it's bound to a protein. And we used purified recombinant protein for all three with a fragment of the LTR. Now, interestingly, this fragment of the LTR was derived from the amplicon that we used in our CHIP assay. And we saw that SAMH1, only the wild-type SAMH1, was able to bind to this fragment of the LTR over salt, although it appears to be salt-sensitive. And then as a control, we took that sequence and we scrambled it, so the GC content is exactly the same. And we did the same anisotropy assay at 50 millimolar and saw absolutely no binding. So it appears that this binding is specific for the LTR, although there's still a lot of work to be done to narrow down where it's binding. So then lastly, a big question is, and Lee always says you can't publish a latency study unless you use primary cells. So we decided to use a Planella CD4 positive TCM model of latency. And to do this, it's pretty straightforward. You isolate naive CD4 T cells from healthy donors, and then you activate them. And then late in activation, you actually infect them with or transduce them, rather, with the GFP virus. And in doing so, and in culturing, you're hoping to produce latently infected CD4 positive T cells. So after we do that, we actually use shRNA specific for SAMH1 and a scramble to knock down SAMH1. So again, we're removing endogenous SAMH1 from these cells, and then we're going to reactivate them using CD3, CD28, and then we'll measure GFP expression. So in doing so, we're hoping to produce a physiologically relevant latency model, remove SAMH1, activate, and then see if there's an effect. And we're able to achieve about 40 to 50% knockdown of SAMH1 in these cells. And what was interesting is when we removed SAMH1 from these latently infected cells, we did see a nice, we did see enhancement of reactivation, indicating that in primary cells, SAMH1 appears to have a role in latency. So to sort of summarize what I've talked about so far, and talk a little bit about future directions. Firstly, SAMH1 is a DHPase. If anyone asks you at any conference for the rest of your life, you can say that primarily restricts AHMB1 infection in myeloid lineage and quiescent CD4 T cells through DHP depletion. Now, even though we know this, the complex control of this protein is still under debate. How it's phosphorylated, the binding sites of the cyclin binding proteins. Also, there may be an additional mechanism of control that we don't know yet. Every week, new papers come out. So there's still so much to explore about SAMH1's ability to restrict HIV infection through its DNTP regulation. I've also shown you that SAMH1 is a DNA binding protein that is capable of binding HWV1 DNA. Now, even though I've shown you this, we still have a long way to go to characterize this binding. We don't know, so far the literature has told us, has given evidence that the binding is not sequence specific, but I have shown you evidence that it appears to be specific. Could it be structure-based? We don't know at this point. We also don't know where SAMH1 is binding on the LTR, so the awesome grad students that are taking over my project will have a lot of fun doing some hydroxyl radical footprinting. Glad I'm not you. Also, I've shown you that SAMH1 does suppress LTR-driven gene expression. This is a preliminary study. There are no other groups that have shown this at this point, and it does need to be further characterized. Is this specific to HIV-1 or retroviruses or only human retroviruses? We have some preliminary data, but we're still trying to confirm. Whether it's this is also due to direct binding is still, even though I've shown you evidence, there's still a chance that SAMH1 might be modulating the innate immune response, specifically through the NF-kappa B pathway, and that could be responsible for this suppression. And also I've shown you that SAMH1 is a player in HWV1 latency. There are a lot of host proteins that are involved in latency, and although we don't at all hypothesize that SAMH1 is the most important or the only, we still need to do experiments in primary cells that have been isolated from infected patients to fully confirm this statement. So I knew I should have worn waterproof mascara. I'm sorry. I have a lot of people to thank, and I'm a very visual person, so there's gonna be a lot of photos, and I'm sorry if you don't like your photo. Firstly, before I even start, I want to thank my collaborators, Green, Monsieur Forsyth, who's also on my committee, and especially Alex, who helped design and helped me do and helped me analyze all my anisotropy assays. There was one day when I added the wrong volume of water to like 75 tubes, and it was my nightmare come true, and Alice just sat down next to me and replaced all the tubes for me while I might have openly cried. And I could have done it without her. I also want to thank Dr. Yunt for his work on, well, for his work in a lot of things. But specifically, he was responsible for the IV of the influenza Sendai virus infections in the first part of the story. And he's always been a joy. He always comes and says hi when he's in the basement. It makes me feel really special. So thank you for all that you've done for me. Young Jean at Yale is responsible, actually, his two graduate students, Kristen and Olga, are responsible for all the purified protein. Lee likes to point out that they purify protein for me in 72 hours, which is pretty amazing. And I get it within 72 hours, so I'm really grateful for them. And then Beck Kim has done all of our DNTPase assays, which means I didn't have to do them, so I was really grateful. I also really want to thank my committee, Karine, Irina, and Jesse, who just have been amazing mentors for me the last 4 years. I feel so grateful that I was able to have you on my committee. And you have about 5 minutes to bribe them, so you know what they look like now. I honestly felt so supported the last 4 years by them, even when they were asking me really hard questions, because I always knew they were asking me hard questions to make me a better scientist. And Frederica, thank you so much for being here and for being my graduate student representative. I hope you like the talk. Okay, so Lee, I love this photo. I don't know if you do, but first and foremost, I want to thank Dr. Wu for giving me a chance 4 years ago. I was very overeager, and I don't know if I would have given a chance to someone who was bouncing in their seat during our meeting, but everything I'm able to accomplish is because of your support. I don't know what many other PIs would have helped me publish a paper over the course of a year through revisions, who stuck with it just so that I could have that high impact publication. A few months ago, when I told him I was going to have a major surgery 6 weeks before my defense, his first comment out of his mouth was, "Just take care of yourself. Take time off. You're going to be okay." And the last 6 weeks have been really hard, and every time I go to him, he's just been so great and supportive. So thank you, Dr. Wu. This was the first photo I ever took with my lab. It was a lot smaller back then, but I just want to point out Suresh De Silva, poor guy. He left probably because he was the one who had to train me when I first joined the lab. He was very amazingly patient. He taught me how to transpect when I thought I knew how to do better than him. And he taught me how to be patient and how to give a good presentation and how to reread an email before you send it. And Fei, who was a graduate student in the lab when I joined, she really super helped me get used to being in a lab and being in the Woo lab and being a team player. And Rebecca too. Rebecca was in the lab, but all three of them left, and our lab got a little bit bigger. And we got Karthik, who was in my office with me and had to put up with a lot of my talking, but was a great friend and mentor. And Chin, Raj, Wushun, and Serena all joined the lab really quickly. And they've all been a huge part of my life. And this is the lab today. It's gigantic. Pretty soon I'll have to Photoshop me out though. We got Shuliang, Sun-hee, who, I mean, Sun-hee's amazing. There she is. Everyone should literally applaud her. Because yesterday she prepped 50 samples for flow and they were beautiful. She does so much work for all of us in the lab. We couldn't do anything without you. And then Tyler and Victoria are the newest members of the lab. We're so happy to have them. And you guys are going to do great. And I just want to point out, Raj loves taking up photos, so I'll cherish these forever. So for those of you who have never been to the basement, I feel so sorry for you because it is the most joyful, amazing, fun-filled place to work. You can go from talking about your weekend to talking really serious about science to talking about like what you eat, because I always want to know what people are eating. And I just want to point out a few people in these pictures that have been just so important to my life. The first is Alice, who, when she gets stressed, she bakes, and that really helps me. I already talked about her science, but as a friend, as a person, you couldn't be luckier. Corinne, I always call Corinne my mentor because that's what she is, but she's also a best friend. She's trained me and challenged me, and sometimes says stuff that's hard for me to hear, but I'm always grateful. She manages the lab amazing and always makes it feel like home. So thank you, Corinne. This is Jacob. This is my favorite photo of Jacob Alstein. I've never laughed so much in my life except for when I'm working with Jacob. It helps that he gets along with my husband. Jacob is not only an amazing scientist, but he's a great person. And he always tells me, he always looks at me when I'm stressed and says, Jenna, you're good at question. So thank you, Jacob. And then I also really want to thank my cohort. I don't have a picture of all of us, but we were just like a bunch of nerds that came in at the same time. And my goodness, I wouldn't have been able to get through anything without their support. It's been incredible to know them. The two people that I really want to point out, though, who have helped me the most are Lindsay Solder, who's actually not in the photo, but she's right there, so you know what she looks like. Our dinners, our lunches, the support, everything you've done for me leading up to this day. And then Brent Simpson, who has been there when studying for Joe Kritzky's class, when things went from this to this very quickly. Always there to help, always there the day before big events to cheer me on, always there to knit me really cute things. Thank you, Brent. So if you were curious, who though is number one responsible for turning this into this, it's my parents, my mom and my dad. That's very good, Janine. My parents aren't they so cute? My parents are, they're amazing, and even though they don't exactly understand what I do, they're always rooting for me. So many times I get a call in the middle of the day, telling me that everything's going to be okay, that everything's going to be great, and they celebrate better than anyone in the world. Also, the rest of my family, my sister Christina, my brother Joe, my new brother-in-law Brian. I want to thank you guys. They're actually on the webcam right now. I love you all so much. And then my in-laws, Kevin and Amy, who are here. My sister-in-law, Frances, and my brother-in-law, Andrew, and their babies. There's actually one more, but I don't have a photo of Catherine. Thank you all so much for your love and support. I don't know how I'll ever thank you. And then lastly, sorry, just felt like I really want to take this on. To the person that I like so much, I married him not once, not twice, but 3 times in grad school. My husband Wendell, my best friend. I really think when someone gets their PhD, their spouse should like get an honorary degree. All of you who are married in grad school or dating in grad school know it takes a village. And I've laughed along the whole way because he has just kept me on my toes and going. And in the morning, sometimes he gets me dressed while I'm still asleep because I really don't want to go to work when I'm really tired. So thank you, my love. It definitely takes a village, and I want to thank you all so much for coming and for supporting me through the last 5 years. And I can't wait to see what's next. So if you have any questions, I'm ready for them.
Thank you, Jenna. And before we start questioning, I actually want to save the best at the end because Jenna already talked about this two major projects that she led as the first author. Now you understand how fun to work with Jenna. Actually, she participated in other two studies. We just recently published with the Yangzheng Lab in Nature Communication, and also PNAS paper we recently published as a team. Both projects, Jenna made a very important contribution in addition to her own two first author research article. And also I want to share with you one review article she wrote within 13 days. I can't remember the exact time. Because we were invited to publish this review article, but very, very tight deadline because they have this special topic in Journal of Frontiers. Microbiome immunology. So Jenna was able to lead that and finish that. So now open to public questions and receive committee later. Thank you.
Yes.
Were you surprised that the phosphorylation you didn't have activity in it?
Yeah.
Usually think that that's an activity.
I was. And would you like me to hypothesize why?
Sure.
So one of the things that a few structure papers have shown is that when SAMH1 is phosphorylated, it falls apart into monomers. And it's also been shown that SAMH1 only binds nucleic acids as monomers. So if we have a phosphoablative, the hypothesis is that in cells, it's a more stable tetramer because it's not being phosphorylated, and that might prevent its binding to nucleic acids. Now, why we don't see it binding in vitro, I'm still trying to figure that out because we don't add any DNTPs in our in vitro binding assays. So whether it's a monomer or tetramer isn't as important. But that's sort of our thought process now as to why the phosphoablative mutant isn't suppressing in cells. Yes?
Is SAMHD1 known to bind to any other viral UTRs?
So not bind. I haven't seen any, and we haven't done any experiments of it binding. But I do have a piece of data that shows that SAMH1 is able to suppress HTLV1 LTR driven gene expression, but not MLV1 driven gene expression. So it could be binding. We haven't done those assays yet, though. Yes.
So piggybacking on that, are there any proteins that SAMHD1 interacts with in the cell to be binding those LTRs and just bringing SAMHD1 along?
So I don't have any identified, but absolutely that could be happening. So when we do our binding assays, we can't say for certain. We IP with an anti-HA antibody, but I mean, it could also be a protein. So we can't say for certain that it's a direct binding.
Can you do gel shift assays in vitro and just ask if just purified SAMHD1 binds?
I have done EMSA assays and then the fluorescence, so we know it's binding, but it could be non-specific.
Good question. Actually, we just talked about this because we published last week the SAMH1 binds to NF-kappa B, the most important transcription factor. Jenna already showed the NF-kappa B 2 sites in LTR. Maybe this is a complex, but this is going to be a new question for new students.
Yeah.
Good question.
Thank you.
Any other questions?
Come on, any more? Dig into
So this
it.
is very unusual. Jenna always smiles, no matter how difficult the project could be, because she's very popular even for anonymous review. Her Nature Medicine paper and the Journal of Virology paper both got 4 reviews. I don't know why, it's just so popular. Every time we got 4 reviews, and Jenna was very bold to face all the challenges.
So lucky.
All right, so thank you everybody for coming.
Yeah.
I love you. I'll call you later. Was it actually good?